And now we are delighted to have Dr. Timothy Kiefer, Director of the UBC Life Sciences Institute, here for this evening. Please join me in giving him a very warm welcome. Thank you.
Thanks for that introduction, and particularly thank you for that promotion to the Director of the Institute. So indeed, my name's Tim Kiefer. Welcome. I'm not the Director of the Institute. Dr. Peter Collis is, and unfortunately he couldn't be here tonight. But I am the leader of the Diabetes Research Group in the Life Sciences Institute. And I would encourage you to check out our website to find out a little bit more about what we're doing. This is one of a series of events that's being organized by the Life Sciences Institute, and we'll tell you a little bit more about some of the upcoming ones at the end. Tonight you're going to hear some great speakers talking about drugs, diet, and genes, personal approaches to treat diabetes and obesity. Some other housekeeping things. First of all, I'd like to acknowledge the Barber Learning Center, who is filming this tonight, but don't worry, none of you will be captured in the film, only the speakers will. We'd like to thank the Vancouver Public Library for hosting this, BC Diabetes for supporting this. You're going to hear a lot more about that tonight. And as I mentioned, this is being organized by the Life Sciences Institute, and I want to start this session before I introduce the speakers, by telling you a little bit about the Life Sciences Institute and a little bit about diabetes to set the stage for the next speakers. So the agenda for tonight, we're going to hear from Jim Johnson talking about insulin, the good, the bad, and the complicated. And then we're going to have a talk by Susanne Clee about do these genes make me look fat? Obviously a talk about genetics aspects of diabetes. Dr. Tom Elliott's going to talk about new drugs in diabetes and give you some ideas about how these drugs work. Or in some cases, as we're going to see, how they don't work and why that might be. And Jerry Klein at the end is going to finish with diabetes management. How do I do what my doctor asked me to do? We will have questions, plenty of time for questions. If you have burning questions during the talk, something you don't understand, feel free to interrupt the speakers. But you'll notice on the tables there's pencils and a piece of paper. Write down questions during the talks, and we'll collect those at the end. We'll have a little bit of a break, and then we'll read out these questions to the speakers to address any questions you may have. So a little bit about our Life Sciences Institute. So this is a building on the main Point Grey campus at the University of British Columbia. It's the largest building on the campus. And the vision statement here is to maintain and improve on its international standing in basic life science research and to become the technological driver for the implementation of personalized medicine in BC. This is a vision that was brought forward by our new director, Dr. Peter Collis, and I think it's really exciting, and you'll get a sample of that from our talks today. So a little bit, some fast facts about the Institute. As I mentioned, it's a very large building. In fact, it houses 800 researchers and has a research budget of $35 million a year. It's pretty impressive. We're very proud to be recognized as #1 in life sciences in Canada. We're very heavy in terms of knowledge translation. So the goal of the Institute is to take the science that's basic research in the labs and to make that into something that can improve the lives of patients suffering from a variety of diseases. So that involves spin-off companies and working heavily with industry to take those things from the lab, from the bench to the bedside. There's already 3 approved medicines that can be tracked to basic research out of UBC. In fact, a new class of drugs that people are taking for diabetes, some of you here might be taking incretin-based drugs. Those were developed from basic research at UBC. And there's 10 currently in clinical trials that have come from basic research out of the Life Sciences Institute. What I also want to highlight is this so-called personalized medicine translation projects. And I believe that's coming up on a slide. I'll highlight some of the different projects in that area. This is a bit of a complicated slide, but again, the concept is to take that knowledge that's being developed in the labs and to use this emerging omics technology, very powerful genomics, sequencing of people's genome, looking at proteomics, looking for biomarkers, and this is based on the realization that diabetes or other diseases isn't the same for each patient. So the idea is, why should we give everybody with diabetes the same drug, for example? We know that some people respond very well to the drugs and some people have adverse events. And our goal is to try to come up with biomarkers, as an example, to predict which patients should benefit from which particular drug, which drug they should avoid. And that's kind of a general theme of this personalized medicine initiative, and hopefully in the future we can use that information to look at someone's genetics and circulating biomarkers and develop preventative medicines in the future. So we think that's an exciting direction we want to head in. So what are some of the projects that we're working on now in this kind of field of personalized medicine? They're listed here. I won't read them all out, but you can see it covers quite a wide swath of areas in medicine. And there will be opportunities at future events to learn more about these, so I encourage you to stay in tune with this series of talks. And of course, this one is focusing on diabetes. So what is diabetes? Again, just a small primer before the speakers get in and tell you about this in more detail. But as you know, the pancreas is where this hormone insulin is produced. It's produced by pancreatic beta cells that are located in the islets of Langerhans. And the job of insulin is to be released into the blood when we have a meal, and it's carried through the bloodstream around the body and allows cells to take up glucose to store it to be used as an energy source. And diabetes results when we don't have enough insulin or we don't have enough insulin action. And as you know, blood sugar rises, and that's what leads to all the debilitating complications of diabetes. Just a few of the staggering statistics. 1 in 12 people in the world now has diabetes. Half of them don't even know they have it. That's why it would be very good if we had predictors, biomarkers, genetic tools to diagnose this quicker. Unfortunately, currently every 7 seconds someone dies of diabetes. So consider during this event, for example, how many people are going to die from diabetes. And a staggering 1 in 9 healthcare dollars is spent on diabetes. In 2014, the expenditure amounts to $612 billion spent on diabetes. So this is the sort of thing that motivates us to come up with better approaches. Here's a world map just showing you how many people around the world have diabetes, and we're up to getting close to 400 million people around the world. I thought this was an interesting slide. If diabetes were a country, it'd be the 3rd biggest in the world. I think we're going to gain, though, because the prediction is from 2013 to 2035 that it's just growing at an alarming rate. So this is really going to break healthcare budgets if we don't do something about this soon, and that's what's motivating scientists at the Life Sciences Institute and elsewhere to work on this disease. And I should mention that you're going to hear a little snippet of some of the research that's being done, but it's much broader than that. Of course, we don't have time to tell you about all the things we're doing with type 1 diabetes, all the various projects we're doing with type 2 diabetes, but there's many of us here from the Life Sciences Institute and elsewhere in Vancouver that are working on diabetes, and we'd be happy to discuss some of those projects with you later.
Come on, Tim, tell us about your plan.
No.
Is it famous type 1 diabetes?
No, we'll leave that for later. One thing I want to point out is that the university provides us with space to do our research, but they don't provide us with money. So we're very fortunate that there's a number of agencies that are all funneling resources into our labs to enable us to do the sort of research that you're going to be hearing about. And another big component is philanthropy. We're very fortunate that some people are donating money to specific projects or just to the university to help support the research that's being undertaken. So have I covered all the important? Yeah. My boss just said I've covered all the important points, and that allows me to move on then and introduce our first speaker, Dr. Jim Johnson. He's an associate professor at UBC in the Life Sciences Institute, a neighbor actually in our lab space, although he's now at Oxford on a sabbatical for a year. and he flew in just to see one of his students graduate and to participate in this event. And he's going to be talking about insulin, the good, the bad, and the complicated. Dr. Johnson.
Thank you, Dr. Kiefer, and thank you all for coming out tonight. As was just mentioned, the Life Sciences Institute tries to take basic science and move it forward in a way that will help with pressing health care problems like diabetes. And as the first speaker, I'm going to start with this kind of makes a ticking sound when it moves. I'm going to start with a really basic story, but I think one that hopefully has some connections to things that everyone here can relate to. And before I start with the basic story, I'm actually going to start by discussing cats, chairs, and balloons. But in all seriousness, I'm going to use these as a metaphor for trying to understand why we do really basic research in the lab, really fundamental research to understand cause and effect. So I actually, I previewed this about a year ago in the mini med school, so let's see how this goes again. All right, so what I want to start the talk with is just the concept of understanding causality, how you know when something really causes something else. Because a lot of things in medicine and in physiology, we know as associations. We know that this is linked to this or associated with that. But it's very difficult to understand what causes what. And we all know that, for example, obesity is linked to type 2 diabetes, but we don't know exactly how these are causally related. And same thing, we know that there are, as I'll get to, there are alterations in, for example, insulin that are linked to obesity, but we don't know whether it's cause or effect. So that will be the topic of my talk today. All right, so this is what I call a correlation experiment. So here you can see that the cat is on top of the chair. But does this mean that the cat is holding the chair up? No, right? Because you have another chair over here without a cat. You can say, oh, well, this one is, the cat is on the chair, so it must be holding it up because there's no cat here. But that's a correlation, right? So that's not a very good experiment. So if you take the cat away, the chair is still there, okay? So it didn't fall over. So it wasn't the cat holding the chair up. All right. Okay, next experiment. So I'm going to take the legs away from the chair, and now we're going to ask, okay, were the legs holding the chair up? Were the legs, are they causal? Are they required to hold the chair up? So we do this experiment, and it goes there. And so what we call this in basic research, we call this a loss of function experiment, where you take something away and you see what happens. And if something changes when you take it away, you knew that that thing was absolutely critical for that event. Okay, and there's another kind of experiment which we call a gain of function experiment. So it's I won't talk too much about these types of experiments today, but for example, you can imagine if I attached a balloon, this of course wouldn't work because the balloon isn't big enough, but if I attached a balloon here, you could imagine that the balloon could hold the chair up, but that's not how it always works. So you could make a balloon hold a chair up, but it's really the legs that are required. Anyway, as I alluded to a minute ago, there's this whole mishmash of factors that are all associated with each other. So we know, for example, that obesity is associated with changes in insulin, and we also know that changes in insulin can be associated with obesity. So, and there's rationale where you can imagine that maybe this causes this, or maybe this causes this. Same thing, there's this concept called insulin resistance, where the insulin doesn't work properly, and maybe the insulin resistance causes obesity, maybe the obesity causes insulin resistance. Maybe the alterations in insulin cause insulin resistance, or maybe vice versa. These things all sort of happen at the same time, and what you need basic research for is to try to understand, well, which is actually causing which. And of course, out of all of these factors comes type 2 diabetes. So what we decided to do in the lab was actually see what would happen to all of these things when we reduced insulin itself. And actually, this hadn't really been done. It had been done with some drugs, but it hadn't been done at what's called sort of a molecular level. So we actually removed some of the insulin from our experimental system. So the reason we did this is there's sort of a classical model. And I was sort of bothered by it. We wanted to sort of test the hypothesis of causality. So that the sort of classical model teaches us that if you have sort of an unhealthy diet, in the case of a mouse, for example, if you give them diets high in fat, they will get obese. And then it's thought that that leads to something called insulin resistance, where the insulin doesn't work as well. And then it's thought that leads to a compensatory elevation in insulin. So it's thought that the insulin rises up to help out the insulin resistance. And then eventually the beta cells run out of insulin and you get type 2 diabetes. So this is sort of what's taught in medical schools and generally in physiology. But there were some suggestions that maybe the high levels of insulin might actually be sort of up on top here and potentially causing some of the obesity. So this is just another way of saying that. And in fact, a long time ago, a group led by Jesse Roth had sort of suggested that the hyperinsulinemia, maybe it's the cart, maybe it's the horse, who knows. So there were a bunch of associations, and they thought that maybe they would look at that. And then there were some clinical studies, both in humans and in animal models, in these very fat rats, that suggested that if you reduced insulin, you would actually cause them to lose weight. So this is a little bit backwards from what we're normally used to thinking, because we always think of, especially in the context of diabetes, as insulin as being good. But this suggested maybe sometimes there's too much insulin. So that was one idea. And then there are, I don't know if any of you follow the diet books and literature, but there's a huge literature. There's all these books and all these diets that are based on the idea that carbs are what are causing obesity. And we wondered if there was some sort of merit to this idea. Now, all of these, whether it's the paleo and low carb, I just figured out what many of these things are, but there's all these books that have come out, and they say that it's the insulin that is driving the obesity. Now, I actually don't have any particular comment on the diets, and I can get to that in the question and answer period, but basically the whole crux of these authors suggested it's the insulin that causes the obesity. So I didn't know if that was the case. And then, and this is an example from a mouse experiment, but these mice here were fed a big, like a sort of a bad diet. So they're fed a high-fat diet. They got quite fat. These mice here did what's called an intermittent fasting. So they only ate small, short periods of time in the day. And in fact, they were completely or mostly protected from the obesity. And it turns out this fasting protocol kept insulin low. But again, this is an association. So the associations by themselves don't mean that much. So what we decided to do in the lab, and this is something I hope you can appreciate that you can only do in a mouse, is we deleted 3 out of the 4 insulin genes that they have, or the insulin alleles that they have. So we took a mouse, we got rid of all of its insulin too, and there's genetic engineering you can do to delete genes. It's actually pretty cool. And we got rid of all of one of the insulin, and we either had 2 copies of this other insulin or 1 copy. So we made a mouse that was genetically incapable of having high insulin levels on a high-fat diet. So you can see this mouse, which is our control mouse, it gets high insulin. But the mouse with less insulin genes actually can't keep its insulin high. So this was the first time we were able to distinguish the effects of insulin itself. This is that loss of function experiment that I was suggesting you could do. So now we can ask, is insulin required for weight gain in a mammal? And we're all mammals. Okay, so here's the answer. It is. So the control mice in pink got fat when we gave them the unhealthy diet, and the mice with reduced insulin gene dosage were protected from the diet-induced obesity. And that means that the insulin the high levels of insulin must have been required. And I don't need to get too much into the details, but it was because they actually burned more energy in their fat. And we figured out a bit how that happens, and you don't need to know the details that much, but it turned on a gene that actually wastes energy in fat in favor of heat. What's that? It would be. It would be. That's true. People are working on that. Yeah. So I have one more little experiment to show you, because we redid this experiment. That other one was male mice, and we did another experiment here now with female mice. The female mice in this particular experiment were a little bit different because the insulin stayed low, but then it went back up again. So this allowed us to test the hypothesis that a transient reduction in insulin might have sustained effects on weight gain. So sure enough, in this case, and you can imagine this as sort of teenage girl mouse. So these mice that had low insulin through their sort of growth period years, even after the insulin was the same, they're still lighter than their controls. So that suggests that there are effects if at the right time in sort of development you can have major changes. So this tells us that the insulin can help drive the obesity. Now there's probably lots of loops in here, and we're working really hard to try to figure out more of the causal effects. But we think that the insulin itself is playing an important role. So I'll just finish with my conclusions. One of the key things is to say that we really need to do a lot more research. So more research is really desperately needed. For example, even though the people who've written all those books sort of think they know the relationship between the different diets and the fasting insulin, I don't think we really understand that well. And most importantly, and as was mentioned with the personalized medicine, we don't know whether each person would respond with high levels of insulin to each different diets. You know, we have Canada-wide recommendations for diets, but it's quite clear that across Canada we have a lot of different types of people, and we know that people respond differently to different diets. Do their insulin levels change differently to different diets? In fact, no real study has linked the genetics of specific, different people's genetics to the response to insulin to different types of nutrients. And this is something that we're hoping to do at the Life Sciences Institute. But you probably say, and these are what we would call gene-environment interactions, which Dr. Clee will talk about more. But in the meantime, I mean, I can make some suggestions. I think it is wise to reduce food intake, especially sort of empty calories and junk food and whatnot. Things you can do to reduce your fasting insulin if you're a healthy person. Otherwise, would be a really good idea. I also think that last experiment tells us that childhood and adolescent nutrition is incredibly important because, it's easy to tell somebody when they're 30 or 40, oh, you should change your diet, but we all know that's a real sort of uphill battle, and probably a lot of what happens in terms of our ability to deal with different diets is really dictated when we're a young child. Even in utero, there are a lot of sustained effects that can happen in the physiological system. So I think there really needs to be an emphasis on early childhood nutrition. We still don't really know what kids exactly should be eating precisely. And then I do want to say, because I do get asked this all the time, the experiments I showed you, of course, they're animal models, not people. And they're in non-diabetic animal models. So I would not want to suggest at any point in time that people who have diabetes should reduce their insulin. First of all, no, a doctor shouldn't listen to me with advice anyway. But you should not. Nobody should be stopping taking their insulin because of these studies. These are animal model studies on normal glycemic people. But what it does sort of tell us is that insulin isn't just good or bad. That we all have a certain level of insulin, and of course, if it drops below a certain level, you get diabetes. But if you have a very unhealthy diet, you may actually have more insulin than you need, and that can have deleterious consequences too. So, we shouldn't think of things as either good or bad. I would say that they're typically complicated. So that was my last slide. I think I'll hand it back to Tim, who will introduce the next person. Thanks.
Thanks, Jim. That was a great talk and really highlights the need for personalized medicine, right? Insulin can be good, bad, and complicated, indeed. Remember, if you have questions for Dr. Johnson, write them down on a piece of paper, and we'll come around and collect those later and ask them towards the end of the evening. So next up is Dr. Susanne Clee. So she is also a member of the Diabetes Research Group and in the Life Sciences institute that I introduce you to. And her area of expertise is in genetics, and the title of her talk is, Do These Genes Make Me Fat? Dr. Clee.
Well, thank you, Dr. Kiefer, and thank you, everyone, for coming. It's great to see such a good turnout here tonight. So, I'm not going to talk specifically about these, but I'm going to talk about something that's one of the biggest risk factors for type 2 diabetes, and that's obesity. So here in Canada, as I'm sure you're all aware with what seems like more and more frequent news reports, we're facing an obesity epidemic. So dating back even to 2004, it's been estimated that over 60% of the country is actually overweight or obese. And what's remarkable about that is it's being seen even in kids as young as 2. And what's really scary about that is that these individuals are at increased lifelong risk for all the complications of obesity that I'll talk about. So you've been hearing about the obesity epidemic, but what does that really mean? Well, in the year 2000, it was estimated that there were about 1.2 million Canadians that were obese. By the end of this decade, that's estimated to, or projected to triple. So over 3.7 million Canadians by the end of the decade will be obese, with even more of them being overweight. And why that's a huge problem is that not only due to the health risks, but projections are anywhere between about $4.5 and $7 billion that we spend per year just dealing with the health costs of obesity. And that doesn't include all the lost productivity and the associated diseases like diabetes. That's just due to the obesity itself. So I've mentioned some of the health complications, and I'm sure many of you are aware that obesity is a big risk factor for type 2 diabetes, but it's not just diabetes. Obesity is also a risk factor for high blood pressure, heart disease, stroke, many different forms of cancer, gallbladder disease, arthritis, and the list goes on. Actually, most common chronic diseases have at least some risk associated with obesity. And in Canada, it's estimated that obesity itself directly causes at least 8,500 deaths a year. So what causes obesity? Why are we sort of facing this problem? And the very simple answer can be traced to what our physics tells us, and that's going way back there that energy can't be made. So when energy in and energy that you're expending through just maintaining your body plus activity, when those two are equal, your body weight is stable. But when those get out of whack, so when you're eating more than you're expending and you have an excess of calories, that causes an increase in body weight. So that's the nice sort of simple explanation. But really the picture is far more complicated than that. So there are many, many, many factors that are all contributing to the obesity epidemic. Amongst these, obviously, environmental changes, dietary changes, lifestyle changes, the list goes on, and this is really only a partial list. So what can be seen from these, though, is that biological factors actually play a role in a lot of these different things. So our biology affects how we smell and taste our food. It affects actually what kind of food we prefer. Are we really into sweets? Are we into more fatty, salty foods? Factors affecting how we digest and absorb the calories that we eat. Obviously, our metabolism, exercise, what type of exercise we're actually good at, how much we benefit from different types of exercise. All of those have biological or genetic factors that contribute to them. So that leads to the question then, really, are there genes that can cause obesity? So this is something that researchers have been interested in for decades. And the first real example that, yes, a gene can cause obesity was originally discovered back in the late 1940s when a mouse that was used and being studied developed a spontaneous mutation. So all of a sudden, researchers noticed that some of their mice got really, really fat. And so they named this group of mice the obese mice. And it took, at the time, decades to finally figure out what the gene was. But back in the mid-1990s, finally the gene that causes these mice to get fat was discovered, and that was a gene called leptin. So what leptin is it's actually a hormone that regulates food intake and energy expenditure. So this is one of the first really clear examples that those factors aren't just all willpower or things that you do yourself. They're actually biological hormones in your body that can control your food intake and your energy expenditure. So, that's fine. This is something that happens in mice, but is it relevant to people? Well, yes, it is. So shortly after the mutation in the mouse was identified, people started screening children and those that they had in clinical practice that had very severe obesity. And some of them were indeed found to have mutations in the leptin gene. So this is a child here who, at 3 years old, weighed about 42 kilograms, which is about 90 pounds. And that child was found to have a mutation in the leptin gene. Now, what's really good about this, and what leads into personalized medicine, is that we now know we can give people with leptin deficiency the hormone back. So this is the same child at age 7 years old, who at that point only weighs 32 kilograms, or about 70 pounds. So this is one example of how knowing the genetic cause of a disease can really inform treatment. Now, what we were hoping when this gene was discovered was that it was going to be able to treat everyone with obesity. But like Dr. Johnson was mentioning, just like with type 2 diabetes, the hormone insulin doesn't work as well. In this case, what was found is in most obese people, they become resistant to the effects of this hormone leptin, so it doesn't work as well in them. But for the subset of patients that have obesity due to leptin deficiency, this has been a tremendous cure. So that's great. That's one example of a gene causing obesity. But unfortunately for most of us, that's not the standard. So very few cases of obesity, and certainly none of the differences in body weight in this room, are likely due to mutations in a gene like leptin. But that said, there's still a very strong genetic component to obesity. So this is data from a study of twins, where people evaluated what was causing their differences in body weight. And what was found is that about 65% of the differences in body weight between twins were due to their genetics, whereas only about 35% of it was actually due to their environmental differences, so their lifestyles, their dietary habits, things like that. And this genetic contribution to obesity or body weight is actually very similar to what we see for height. And while most people wouldn't be surprised to think that genes affect your height, most people don't tend to think of the same thing for your body weight. But in fact, the genetic component is just as strong for weight as it is for height. And if you think about that, that really makes sense, because the drive to eat and drink are essential for survival. If you don't get hungry when you're low on nutrients, you're not going to do too well. So that sort of ingrained, or that drive to eat, is a really strongly genetically determined behavior. The problem is when calories are abundant, that's when things have started to get out of whack. So I said that common obesity was far more complex, and it is probably one of the most complicated genetic diseases out there. So with all these new omics technologies that you'll be hearing about and that are driving this personalized medicine initiatives, what's been shown for obesity is that there are probably actually likely thousands of different genetic variants out there that contribute to obesity. I've seen estimates of up to over 3,000 potential genes that could contribute. And with these new omics technologies, recent advances have managed to find about 60 to 100 genes are the current estimates. But those took studies of over 300,000 people just to find those 60 to 100. And of those, there's one that has a pretty strong effect, but most of them are all at the levels where we can barely detect them. So what's been shown is that each of these different genetic factors affects your body weight by maybe about 500 grams, or about a pound. So what determines our body weight overall is the whole combination of all these genes we get. And in every gene, there are some that will increase, one form that will be associated with a bit higher body weight and another form that's associated with a bit lower body weight. And so it's the sort of net effects of all the good and the bad gene versions that you have that ultimately determine your genetic components. So if each gene doesn't contribute very much, what does that mean? Well, what the researchers have shown is that when you do add them all up, it actually can have a big effect. So this is data from a bit earlier in some of those studies, and what researchers did is looked at where they knew the genes, people that had over 38 bad versions of the genes they knew about, compared to people that had less than 21 of the bad versions. And what they were able to show is that just that difference actually amounted to about a 7-pound difference in body weight. So they really do all add up over time, but each one themselves just has a very, very small effect. To add on to that, as Dr. Johnson mentioned, there's things called gene-environment interactions. So you can imagine something that affected how well you metabolize a high-fat diet. Well, if you're not eating a high-fat diet, then that doesn't really affect you or have much influence on your body weight. In contrast, if you're eating at McDonald's every day, that may have a bigger effect. And so what this graph shows is that these interactions modulate. So if you have a low genetic susceptibility, then whether you have a low-risk lifestyle environment or a high-risk, it doesn't make that big of a difference on your body weight. But if you have a high genetic susceptibility, then the effects of that high-risk environment can be magnified. And so when we're trying to look across people, like if I wanted to understand what caused everyone's body weight in this room to be different, we'd have to factor in all these different environmental things, diets, lifestyles, you name it, across everyone's lifetime, and then try and pick apart the genetics. And so that's just really complicated, and that's what's making it so difficult to find genes in people. So why are we bothering? If it's so difficult, why would we want to bother with this? Well, obesity is really a global health problem. It's not just Canada. So right now, there are over 2.1 billion people in the world that are either overweight or obese, and that's 30% of the global population, and it's estimated to reach about 50% of the global population by 2030. And then if you can imagine all of the complications like diabetes that will result from that, that may drive Dr. Kiefer's graph even higher. So why we want to understand the genetics is, like Dr. Johnson was mentioning, is to understand what's causing the disease. Then we can find new treatments that are based on actually fixing what's causing the problem, not just trying to deal with the symptoms. And the other thing is trying to figure out which diets or what treatments are going to work better for what people. And that's really the goal of personalized medicine. So in my lab, because all of these things are so difficult to control in people, our work, we use mice, because then we can actually control a lot of these things or keep them constant. So if we keep their diets constant and they're having the same size cages to run around in and all those things, then that makes finding the individual genes much easier. So I'm going to give you two really quick snippets of some of our work that we're doing right now. So here's our good old friend, the OB mouse again. But what was found is that if you have mice that have that same mutation but are a different strain. So different strains of mice are kind of like different breeds of dogs. You can think of them like that. They're still mice, but they're different from one another. So when mice on a strain called BTBR have this obese mutation, they actually get fatter and they get really severely diabetic. And so in prior work that I've done, we used that fact and have actually found two different genes that affect how diabetic they get. But what we're working on in my lab right now is trying to find the genes that affect why they get fatter. So we started off with the whole genome in mice that we had to screen through, and that's about 3 billion base pairs. So you can think of a base pair as like an individual letter in your DNA. So the whole mouse genome is about 300 billion base pairs, and we've now narrowed that down to only 319,000. but that's actually a 99.99% reduction. So we're getting really close, and we've gone from about 30,000 genes in the whole genome down to 2 genes. So what we're working on now is trying to understand which of these 2 genes and how they affect obesity. What we know is that mice, just by changing those 2 genes and making them the BTBR version, the mice get fatter, they eat more, and they're more insulin resistant or more pre-diabetic. The other thing we can do by using mice is actually start testing all these environmental factors. So we know that their changes in food intake are affected by time of day. And this can start to give us clues. We know in humans there's lots of associations between shift workers or people that have off schedules and obesity and metabolic disease. So we think maybe by understanding this, that might be one way we can start to try and understand that problem. We also know that this gene and its effects are modulated by stress. So again, we can start looking at how different genetic factors interact with different environmental conditions to drive disease, in this case obesity. So that's one strain of mice that we work with, but that's one gene. We want to try and understand obesity as a big picture and more genes. So this is a growth curve, or just weekly body weights, of a normal B6 mouse, so not one that has the OB mutation. And like Dr. Johnson mentioned, we feed our mice a high-fat diet. And this high-fat diet is really bad. It's about 60% lard, which is the equivalent of a bacon double cheeseburger. So these mice eat nothing but the equivalent of bacon double cheeseburgers if you throw a little extra sugar into the buns. So if we feed a normal mouse this food, what happens is that over time they get fatter. And they get about 50% heavier than what a normal mouse does eating normal mouse food. But we found a different mouse strain called WSB. So overall, when they're eating normal mouse food, which is kind of like cardboard, lots of fiber and starch and not very much other than the essential nutrients, they're a little bit smaller than a B6 mouse, but that's fine. When we feed them the high-fat diet, they don't gain any extra weight. So they can eat that bacon double cheeseburger diet their whole lives, and they seem to be protected from all the forms of metabolic disease. So not only that, we've found that they actually eat more. So these mice are eating as much as those mice, and they're moving around less, and they're still not getting fat. And that's because they, again, like Dr. Johnson mentioned, have an increased ability to burn excess energy. So some of the stuff we're working on now is trying to understand and find the genes that cause that. So overall, hopefully tonight you've seen that there is a real genetic predisposition to obesity, that finding these genes is challenging, but it's really important. But like Dr. Johnson said, that doesn't mean that we don't want to leave you with the impression that diet and exercise aren't important. Our genes may determine, and hey, I'll never look like a supermodel, but we can all be healthier with eating better and exercising more. So with that, I will leave it and pass it back.
Wow, that's exciting. Can you imagine if Dr. Clee finds out the secret of those WSB mice? There'll be lineups for those double cheeseburgers at every fast food restaurant around. So in the meantime, while we're working on all this exciting research to come up with new strategies to treat diabetes and obesity, how are we treating diabetes right now? We're very happy that Dr. Tom Elliott took time out of his very busy schedule to tell us about new drugs in diabetes. Dr. Elliott's the medical director of BC Diabetes that I introduced you to earlier, and an associate professor at UBC. Dr. Elliott.
It's great to be here. My busy schedule this afternoon included a dance lesson at 827 Granville. I'm not that busy that I can't enjoy moving. I'm excited to be here. It's so great to be mixing with real scientists, bench scientists, people who are getting down right to the, not just molecules, to the DNA to figure out why everything happens. I'm I'm a doctor. I'm a diabetes specialist. I spend most of my time seeing patients trying to figure out how to lower their blood sugar or lower their blood pressure or lower their cholesterol or lower their weight. It's exciting to hear all these ideas. And actually, having heard Jim speak, saying that, that insulin may not be good, since I spend most of my time encouraging people to take insulin, I feel like changing my title to Insulin is Good. Hearing Susie talk about rats and how well she can control their environment, I might have changed my title to I wish my patient were rats. And they would actually do what I suggest or what I want them to do. And then she even talked about rats' normal diet being cardboard. Well, when I'm sort of joking with my patient, I want them to eat less and exercise more. I tell them that all I want you to do is jog and eat cardboard. But that's actually what your rats are doing. And then I asked Tim Kiefer, I said, Tim, how many base pairs are there in the human genome? Because Susie had told us there are 3 billion. Well, Tim said, I think it's about the same, Tom. Because they're mammals, we're mammals. We all started out one way. So the crazy thing is we are actually really close to rats or mice. That's exciting. And one other thing occurred to me, what got me into science in the first place? It was Charles Darwin and evolution. The glorious idea that, in my humble opinion, everything can be explained or understood with science. So I could have made the title of my talk, Science is Glorious. Science is glorious. It's so exciting. It's what gets me out of bed in the morning. Now, I only have 10 minutes to talk, but I don't have much to say. So, hey? Only 6 now. Okay, so what causes diabetes? It's pretty simple. There's not enough insulin. So if we were going to do a Jim Johnson experiment, you take something away. We know if you take insulin away, people die. That's called type 1 diabetes. And Tim Kiefer is already famous. He's going to become even more famous when he figures out how to grow stem cells into insulin-secreting cells and inject them into humans who are lacking insulin completely. That's called a cure for type 1 diabetes. I'm losing my track here. Insulin is good. Oh, yeah, so a subtraction experiment, take insulin away, people die. What if you add something and they live? That's called an additional experiment. And that was the beautiful thing that Banting and Best did in 1922 when they discovered insulin. They gave insulin to people, and instead of everybody being dead within a year, nearly everybody was still alive 10 years later. So that's a sort of exciting thing. And that's why I want my title to be Insulin is Good. I certainly agree with Jim that too much insulin can be a bad thing. But when you don't have enough insulin, when your blood sugar rises, you want more insulin or you want more insulin action. Now, the main reason that type 2 diabetes is so common, it's 90% of all diabetics. So if 8% of Canadians have diabetes, then 7.2% of Canadians have type 2 diabetes. That's mostly because their bodies run out of insulin as they get older. The cells that make insulin die off. Natural cell death. You get wrinkles, your hair goes grey, you get diabetes. That's getting older. So it would be great if we could figure out how to stop that happening. So there we are, not enough insulin. It's either because you're getting older, type 2 diabetes, or your insulin cells get destroyed. That's type 1 diabetes. What about insulin not working well enough? They're the obese rats that Susie talked about. And then to balance that, Jim said, well, you know what? If you lower insulin levels in non-diabetic rats, they lose weight. And if you particularly lower it during adolescence, they don't get fat even though their levels are the same as other people later. So it's very confusing and very interesting. The bottom line is that one of the reasons there's not enough insulin is that the insulin isn't working properly. The commonest one is overweight. Slightly less common is underfit. So the fitter a patient, the better their insulin works. If you get an obese patient to lose weight, their insulin starts working better, their blood sugar goes down. Illness and stress makes insulin not work as well because it causes the levels of hormones that oppose insulin to be higher in the blood. Flight or fright, getting scared, raises our adrenaline level. The higher the adrenaline, the less well insulin works. Our bodies release cortisol from our adrenal gland. That makes insulin not work as well. Our bodies release growth hormone as well. And there's another hormone called glucagon, also made in the pancreas, that is an anti-insulin. Sometimes you get put on medication that's going to make your own insulin work less well, or the insulin you're taking work less well. The common one is prednisone, which is given for arthritis and asthma and things like that. And then people going through chemotherapy may find that the drugs are lowering their insulin effect. And finally, lots of genetic causes. So I'm seeing patients. What is my job? My job are these 5 things. The first thing is to educate my patient. If I can explain to my patient that the leaner and the fitter they get, the more likely it is their blood sugar is going to be controlled, then they're more likely to eat less and exercise more, or as I joked, to jog and eat cardboard. So education is incredibly important. Empowerment. The next thing is the old model where the doctor was the god and told everybody what to do and fixed everybody. That doesn't work. We know that it's all about teamwork, and that most of the therapies for diabetics require the patient to do something. So I'm not going to do everything. I'm going to sit back and say, Mr. Jones, you have diabetes because you chose your parents badly. That's my way of talking genetics and environment. And then I don't say you're overweight or underfit, but I talk about that sort of thing. Most people know that anyway. We've got all these treatments that are effective. Do any of these treatments appeal to you? Do you like the idea of eating less, or do you like the idea of exercising more? Or is there any particular diet you'd like? Is there any particular exercise you like? Maybe reducing stress through meditation or doing a personal development course. Maybe those things are something that you'd like to do. So offer all the treatments that work to the patient and have the patient decide. If the patient decides that's treatments for them, they're much more likely to comply. So that's about empowerment. Perhaps the most important thing that I've learned recently has been the particular role of diabetes case management. Jerry Klein, who's going to speak next, is a diabetes case manager. She knows how to engage, she knows how to do steps 1 and 2, and then to push and prod and to use all the tools at her disposal to get the patient to buy into the plan. So, everybody thinks doctors are overpaid and we don't do anything, and there's some truth in that. But the most important thing I've learned is to get people like Jerry on to work with me so that she can spend lots of time with the patient, get to know the patient well, and find the treatments that patient is going to respond to best. Technology. Technology is a big deal. We've all got our smartphones out. We're all checking the LSI website. We're reading the news feeds. What else are we doing? Sending emails, texts. It's wonderful. The same tools can help improve diabetes. So at BC Diabetes, we're developing apps to improve compliance and adherence, to motivate patients, to give them feedback to see how well they're doing, to give them targets, not just day to day with blood sugar, but week to week with fitness levels, with gradually improving the score that we know is going to get them to target. So technology is a big deal. And finally, research. Better strategies and new drugs. My talk's on new drugs, right? I've got 1 minute left. All the medications we have problems. Metformin is very commonly used. About 20% of people can't take it because of stomach upset. It helps insulin work better. Then there are tablets. By the way, where's the... here it is. We have tablets that squeeze the last drop of insulin out of the body. They work quite well. Unfortunately, they often release too much insulin, which means the patient gets a low blood sugar, they pass out and something bad happens. We have this new class of drug that Tim Kiefer's lab was involved in development, but somehow missed out on, I don't know what happened to it.
Missed out on what?
I don't know, the patent or something you were telling me.
Long story.
Anyway, all these drugs are huge, billion-dollar drugs. This is an expensive drug that works quite well. And then we have insulin. I said that insulin is good. The trouble with insulin is you've got to inject it, and it tends to cause weight gain. Why does it cause weight gain? It's complicated, but one of the main reasons is that people's sugar becomes lower. When it's lower, they're no longer peeing out sugar into the toilet bowl. Peeing out sugar is losing calories. It's like being on a diet. So suddenly someone eats the same. They're no longer losing calories into the toilet. Their weight rises. So insulin has a problem with weight gain and also low sugar. What are the new drugs in diabetes? There is a new drug in diabetes which makes people pee out sugar into the toilet bowl when their sugar is normal. This is called the SGLT2 inhibitor class. There's only one on the market in Canada. It's called canagliflozin. The trade name's Invokana. It's the coolest thing in the last 4 months in diabetes. You end up wasting calories. If on an evolutionary scale, if we start wasting energy, that's not good for our survival. But in our current society, we have an excess of food and energy and money, so we can afford this very wasteful therapy. It causes weight loss. It costs $3 a day. People pee more, and there's an increased risk of thrush, of vaginal infections. So that's this class of drug. The other class of drug that's not quite so new is causing excitement for me from a research point of view, and we'll get to that. These drugs are called GLP-1 agonists. GLP-1 is a gut hormone. There are 2 of them on the market. They're given by injection. They reduce appetite, which you can imagine is a good thing for weight. The stomach is slower to empty. People feel full. It increases the levels of their insulin, and it makes the insulin work better. These drugs are very attractive because they lower sugar and also lower weight, as opposed to insulin, which lowers sugar and causes weight rise. The disadvantage is they're expensive. About 20% of people become nauseated and vomit. And then about 40%, the drug doesn't work very well. So imagine me seeing patients in my office. I'm trying to find a therapy that's going to work, that's going to be inexpensive, and that's not going to make my patient sick. Does this drug fulfill those criteria? It's expensive, number one, so no, the government's not going to pay for it. Even most of the extended medical insurers aren't paying for it. 20% of my patients are going to be harmed by this drug, and then almost half of them it's not going to work. So that doesn't give me a warm and cozy feeling. Well, imagine if we could predict ahead of time who was going to do well with this drug. Not only would we avoid making 20% of the people sick, but we might even avoid giving a useless therapy to patients. Maybe we could persuade government that the 10 to 20% of patients who are going to do really well actually deserve to get this drug through pharmacare. So that's a really exciting idea. And with Tim and some other, and Peter Cullis, the director of the LSI, and others, we're doing an experiment to... Oh, I haven't finished yet. We can go to this. We're doing an experiment to measure proteins in patients' blood, also to measure their DNA, also to measure their microbiome. The microbiome are bacteria in the gut, and these have a big effect on how we respond to many things. So we're going to examine the proteins, the DNA, and the bacteria to see if that will give us a signature of what will predict people who will do well and people who will do badly. So I want to invite those of you who either have diabetes or don't have diabetes to give us a sample of your blood so that we can use you as a control for the experiment. In science, when we do an experiment, we have to, if we're going to intervene with something, we need to know what happens if you do nothing, and that's called a control experiment. So I want you all to sign up as you leave to agree to give me a blood sample later on for this experiment. That's exciting. So it gets me up in the morning. It's hard finding funding, so we also want you to get your checkbooks out as well. And I'm no more deserving than Jim or Susie or Tim in that respect. That's an exciting part of what I do. What else is a new drug in diabetes? Well, surgery for diabetes is actually the single most effective treatment we have for diabetic patients who are obese. How does it work? Well, if you just reduce the size of the stomach with this procedure called laparoscopic banding, people can't eat as much, they end up eating less. They actually feel less full because there's pressure on the stomach. It works reasonably well. The reduction in diabetes rates are about 10%. If we actually remove some of the stomach from the circulation or reroute reroute where food goes through the gut, we get very big changes in diabetes that occur long before the patient has lost weight. And the key to that are changes in hormones that are affecting diabetes. Leptin, like Susie spoke about. Ghrelin is another gut hormone that seems to be involved in satiety and blood glucose metabolism. So this is the most, you can see the numbers. Huge reductions in weight, massive reductions in development of diabetes. If you start with obese people who don't have diabetes, give half of them the operation, half of them not, then there's 50% less diabetes in the people who get the operation. If people already have diabetes and you do the surgery, there's a massive reduction in medication. So this is very exciting. The government doesn't pay for it. It's actually very hard to even buy this treatment in Canada because of the Canada Health Act. But I want to put it out there that this is a wonderful therapy. Personalised medicine, we've already touched on it. Imagine where we could customise the treatment for every patient, not just for diabetes, but for cancer therapy is the most obvious one, for treatments of infection. We have the wherewithal to do this by being very clever with science. In diabetes, ideally we'd have no weight gain, we'd have no low blood sugar, we'd have no nausea, vomiting or diarrhea. We wouldn't cough. Some of our blood pressure medication causes cough. We wouldn't have sore muscles. Some of our cholesterol pills cause sore muscles. So I've already told you about this study. I want your blood to help us with the experiment. And then my last slide is get involved. You're involved actually being here. Volunteer for studies, become a patient, get out your wallet. Thank you.
Thanks very much, Tom. I didn't mention, but on all the tables there's this 1 or 2 page summary to give you a little bit more details about trying to figure out why some patients respond very well to these GLP-1 based drugs and others don't. So this will give you a bit of a summary of the primer of what Dr. Elliott talked about. Okay, so we have one more speaker. Again, remember to be writing down your questions, and then after our next speaker, I'll go around and collect them, and we'll have a panel discussion to address your questions. So Dr. Elliott already introduced our next speaker, Jerry Klein, who's a diabetes educator. And I think Dr. Elliott mentioned to me that all his most challenging patients he directs to Jerry to really figure out how to implement all that advice that he's passing on. So she's going to tell us about diabetes management. How do I do what my doctor asked me to do? Jerry.
Hello, and thank you for being here. I want to change my topic as well. I want to change it to, in the meantime, what are we going to do to be healthy in the meantime? There's great stuff coming, but I don't know how many of you folks live with diabetes. Who lives with diabetes here? Who knows somebody who lives with diabetes? Okay, so most of you. How do you do this? And it's hard. As a by the way, when I completed my nursing diploma the first time 40 years ago, it was almost a guarantee that you would need a kidney. For people with type 1, it was a guarantee you would go blind. People with type 2, it was almost a guarantee you would lose a leg. Through research and efforts of patients, They've managed to reduce that to almost nil. But as you well know, there's still really hard things to do and how to do it. The way you do it is through the A, B, C, D, S's of diabetes. How to be healthy and stay healthy. A little housekeeping. I was the Head Start study coordinator for a study that was funded by Sanofi, and we compared endocrinologist care to my care. I was probably the most unpopular person at the Diamond Center. I am employed by BC Diabetes. References. Everything that I'm talking about can be found on the Canadian Diabetes Association website and BC Diabetes. Free of charge, you can download it. My objectives. Does anybody know what the ABCDESs of diabetes are? Okay, hopefully you'll know at the end of this. Understanding the importance and be aware of how to achieve the targets. A1C. I've put these out on the tables. Folks who live with diabetes all have A1C. Anybody wants to tell me that it's your average blood sugar? Anybody know that is your average blood sugar? Could we be more boring when we say that to you? I mean, really. It drives me crazy. It's more complicated than I'm going to give you, but let me tell you why this is important to you. Within your red blood cell lasts about 3 months, there's a hemoglobin molecule. Hemoglobin molecule carries oxygen and sugar. Your whole brain, your whole body is set up to protect your brain, and your brain actually needs sugar more than it needs oxygen. So your hemoglobin molecule will grab sugar. And if you get too much sugar on that hemoglobin molecule, guess what you don't have enough room for? That's why you care. If you have too much sugar, you're not going to have enough oxygen for your oxygen-sensitive tissue. If you have an A1C, a hemoglobin A1C of 8, that does not mean your average blood sugar is 8. Your average blood sugar is 10. You should have them done every 3 months, and you are the people who need to know it the most. The second one would be the doctor, but you need to know what your A1C is. If your average blood sugar is too high, you can't carry the oxygen, and an A1C of 7, which is considered target, your average blood sugar is 8.2. How to get that? That's the real tricky part. In BC Diabetes, we do what's called purposeful testing. Do not test to find out where you are at. That does not answer a question. Test to answer a question. You test in the morning before you eat to find out if you made enough insulin through the night to turn off the flow of glucogen. You test before each meal and 2 hours after each meal to see if you made enough insulin for your meal times. Note the time that you have eaten in relation to your sugar, and note the times that you have taken your medication. And then take all of that information in and evaluate it with your endocrinologist or your physician or your diabetes educator. Just the number doesn't help. Second thing, B. So we've done the A's, now we're going to the B's. Blood pressure. Blood pressure is the measure of the force on the artery walls. The top reading is the systolic, and it's the most pressure that your arteries would have. And the bottom reading is the diastolic. It's the rest period. And for most people with diabetes, we like them to be under 130/80. You'll notice on the Safeway blood pressure that it says 140/90 sometimes. And those are for folks without diabetes. What we're trying to give you is a little bit of a cushion to make sure that you're not having problems, so that if we lower your target for blood pressure, now you are going to have some readings that are higher, but if most of them are well above 130/80, you need some help. Take it at Safeway. Take it when you're sitting in your doctor's office. Take it when you're staying at home. Don't always just get one reading. Take your blood pressures and bring the numbers into your physician. High blood pressure has very few signs and symptoms until it's too late. You can have damage to your heart, your lung, or your heart, your kidneys, and you can have it for years without knowing it. Know your blood pressure, read your labels, reduce your salt intake, watch your weight, eat your vegetables. Half your plate should be vegetables, really. Those green things. They're not going to kill you. Increase your exercise. Anybody know what aerobic exercise is? Okay, what's aerobic exercise? It is different to every person, so that as long as you're walking or moving and are able to talk but feel a little breathy, for you that may be aerobic. And you will find if you do it often enough, you will increase. Sleep. Interestingly, if you have a lack of sleep, your weight is going to go up, your incidence of type 2 diabetes is going to go up, and your blood pressure is going to go up. So really look at your sleep hygiene. Take medication. If the doctor tells you your blood pressure is too high, take your medication. Get that blood sugar or that blood pressure down. If you're taking insulin, this is a trick question. If you're taking insulin, a cholesterol medication, a blood pressure medication, and an aspirin, which is the most important? Cholesterol, blood pressure, then insulin. So this stuff is important. C, ABC, it's the cholesterol. It's the fatty substance in your body. I will go through them quickly. The LDL is the one to watch. And for people who live with type 2 diabetes, you want your level under 2. Unfortunately, Unfortunately, the cholesterol test is a fasting test, and frequently people don't get it done. Have it done once a year. Know your numbers. If it's above 2, get someone to help you with diet and exercise, and if it doesn't improve in 3 months, get on some type of medication. They matter because you are going to have reduced blood flow because of buildup. Anybody who has had a stroke or understands what it's like to live with a stroke will understand that this is preventative and that it's devastating when it happens. So prevent is the way to go. Targets for triglycerides is under 1.7. Target for HDL, that's the good, helpful type of cholesterol, is 1.5. The most important, though, is the LDL. Keep it under 2. And if you can't do it with diet and exercise, get on a medication. Lifestyle changes, lots of fruits and vegetables. I do have a person who is a vegan with a high cholesterol. Chose the wrong parents. Next time, come back as a Jewish woman. You'll be fine. Really. Reduce the red meats, especially, and pastries, and exercise. Drugs. Everybody thinks that the D is diet, and a lot of the dietitians will probably hate me for it. I have a few patients out in the audience tonight, and I've often said to them, I'm not going to give you a diet, I'm not your mother. So, yeah, half your plate is vegetables, and then eat what you want. You will be prescribed drugs. Take them, and if you're not, tell somebody so that we can get something that's more appropriate for you. My job is not to force you into a diabetic lifestyle. My job is to make the treatment work for you so that your numbers are correct and that it doesn't affect your lifestyle too much. ACE inhibitors are blood pressure pills that will help with your kidneys. Statins will help with the cholesterol, and an ASA is often suggested as a stroke prevention. Exercise and eating. Once again, I'm not going to give you a diet. 5 or more fruits and vegetables, however you wish. Try more veggies than fruit. You may find your blood sugars are better. Exercise. One of the best things I suggest to my patients is a pedometer. Good for you. I actually hate walking. Oh God. But everybody, people who know me, oh, now look what I've done. People who know me will realize that I walk from 41st and Dunbar to 11th and Laurel every day. And then I run a diabetes walk group for the endocrinologists. Few have joined me. And walk the walk. And it's a free service for anyone who lives with diabetes who wants to walk. If you're unable to join us, phone me and I'll talk with you and we talk about diabetes. One of the best things you can do for yourself is walk. 10,000 steps a day, trust me, that's a long way.
How many is Dunbar?
12,000. Yeah. Reduce sitting time. So I walk from 7:30 to 9 o'clock, and then I sit on my keeper until 5 o'clock. And I realized that the walking is very helpful, but sitting is not going to be helpful. So I have to actually get up and move every hour, and I make sure. The physical activity line is supported by your tax dollar. There are a whole bunch of these on your table, and if you phone, they will send you a free pedometer, maybe not as good as mine, but you get one, and they will help you customize some type of exercise program. I have some patients who are unable to walk for a variety of reasons, some who are in wheelchairs, and they've been very assistive, and your tax dollar is going towards it. It says the handle of your recliner does not qualify as an exercise machine. Smoking. The best thing you can do for yourself if you are smoking is to quit. Full stop. It's really hard. I have put some brochures out. If you need some help, I've gone up to the 6th floor of the Diamond Center. There are some excellent smoking cessation nurses who are very supportive. Quit smoking. You can't do anything better for yourself. Stress. I'm less of a fan that stress causes high blood sugar than others. I find that stress tends to cause, well, for me, emotional eating, and I can down 3 boxes of Kraft Dinner in a sitting. So, I have to watch. Yeah, really, I can. I have to watch for stress because I'll be eating the Kraft Dinner. But if you're finding when you're stressed you're eating, you want to develop a different strategy. The other thing I found, and I'm going to share a little bit about my own history, I was on the path to becoming a diabetes educator. It is a 2-year self-study program. I was about 6 months to writing the exam and thought I really knew diabetes. So I was pretty good about it. And then our 6-year-old was diagnosed. That was a big shock. And since that time, he is 24. And as he said, "Why are you speaking? I'm the one who lives with diabetes." Yeah, he knows a lot. You will find that you know way more than you think. So give yourself credit for that. The last thing is become part of the cure. As I said when I started this talk, when I started nursing, and I graduated the first time, I'm really this old, in 1975, and it was 1 in 2 lost their kidney, 100% lost their eyesight. That should not happen. You should not have those. And in fact, through research, they've managed to eliminate these. We can make personalized medicine where you would get one treatment and you would get something else because that's right for you. We need to support this. So become part of the cure.
So we're going to get started now in the question period. If you have questions that you think of, you can bring them up at any point. So the first question is going to be for Jerry. Come on up. So the question is, can you give a description of an ideal diabetic diet? Maybe you can use this microphone there. Why?
There is no such thing as a diabetic diet. You eat what you eat. If you have a diabetic diet, you're probably going to go off it. Everybody knows what the food guide is. Everybody knows that they're to eat more fruits and vegetables, etc. I have one very smart dietitian, and a patient of mine asked her, "Give me a good diabetic diet." And she said to her, "Are apples good?" And the lady said, of course. So is apple pie good? You pretty well know what to eat. People with diabetes don't actually need anything terribly special. They do have to be aware of carbs. They have to watch their cholesterol. Other than that, make sure your calories aren't too high. And exercise so that you can increase your calories if you walk or do some exercise. It's calories in and calories out. So definitely eat and see how your sugars are.
Thanks for that answer. And I just want to come back to Dr. Johnson's talk. He mentioned the Canada Food Guide has one recommendation for everybody, but he also talked about the effects of insulin, and Dr. Elliott talked about how these gut hormones like GLP-1 are different in different patients, and people have different levels. So it's all coming back to this theme of personalized medicine, that one thing doesn't fit for each patient. So I think it's really an exciting time where we now have the tools to look at the genetics and look for biomarkers to try to figure out who's going to respond to one particular medicine or diet or exercise better than another individual. So thanks for that question. This next question, follow-up question? Sure.
I was going to say, would you say the Mediterranean diet is a good basis to build upon?
You really have to try it and then test your A1C, do some purposeful testing, see how it goes to you. Right now we have a one-size-fits-all. We have this little bag of tricks and we throw it at you. What they're trying to do is personalize it so that when you ask that question, I would go on to ask a whole bunch more questions and then say yes or no, because I'd have the answers. As they tease out the genetics, you may actually get a different name for what you've got. And then we'd have a specific treatment. For me, living with a kid who lives with diabetes, that's going to be truly not quite the cure, but it's going to be as close as, that's the best possible care.
Maybe I'll just add on to that. I mean, we really don't have, as researchers, very good answers for these things, because the studies, to do a clinical trial for a drug, and the drug company's going to pay for this, these are millions and millions of dollars. To do the equivalent size experiment in humans, like a clinical trial size experiment for diets, is so expensive. Really the funding would have to come from, it's not going to come from drug companies, they're not selling diets. To do proper evidence-based stuff would have to come from, government or philanthropy. And right now, the power of the study is probably has the ability to answer these questions.
Good, thanks very much. And then just ask the speakers, if possible, to use the microphone to make sure everybody hears you. It's a little short on the cord, but try your best.
At the back, people can hear me, right?
It's for the recording, too.
For the recording as well. Yeah. So now some incretin-based questions that I think Tom can best address. Do you have some ideas as to why a large fraction of people don't respond well to the GLP-1 agonists? What's up with that?
There's a very short answer, and the answer is no. We don't know why. And that's what we want to find out. I want to echo Jim's comment that science is glorious and we do know quite a lot of things, but what we don't know is vast. And I've learnt that, if you don't know, you've got to say you don't know. And that's where we want to go. So this is how you advance medicine, by digging down and finding the right science, doing the right experiment, getting clever people to design the clinical trials very well, making sure that you don't fall in love with your hypothesis. Lots of people have theories. People who write popular books in Costco, there's very little science in those books. They're well marketed. They're persuasive people, but the science is weak.
And they often have an agenda.
Yeah, they're either, it's for money usually. Yeah.
So can I ask a related question, or someone in the audience wants to ask a related question, Tom? So in the same field of drugs, the GLP-1 agonists, of course, are injectable. There's the DP4 inhibitors, which are pills. And I think you noted they're more expensive. So the question is, Are they more effective than the GLP-1 agonists, and are they more commonly prescribed, despite their higher costs?
The GLP-1 agonists, they're the injections. They're 3 times the cost. When they work, they're much more effective. When they don't work, of course, they're less effective. So the DPP-4 inhibitors are pills. They work reliably. They're mildly to moderately effective. So if you want a big effect, don't choose one of those pills. But if you came to me as a patient and your A1C, Jerry mentioned the A1C, if it's only slightly higher than target, then a mildly to moderately effective intervention or drug is what you're going to need. So that might mean walking from 41st and Dunbar to VGH, or it might mean taking a pill.
Thanks, Tom.
Anyone's welcome to join me.
Yeah.
So related to that, I'd like to ask the question, do you have to walk 10,000 steps at one go, or can you walk 2,500 steps 4 times in one day?
Actually, the little bit of research that has been done, it should be about 10 to 11 minutes 3 times a day, rather than the 30 minutes together. If all you can get is 30 minutes and that's what you can carve out, go for it. But if you can divide it up and get to an aerobic level, so that's why I say 11 minutes, do that 3 times a day, and that might be getting off the bus early. It might be parking rather than in the $20 space under the Diamond Center up at 19th, where it's free, by the way. You'll get 11 minutes each way. Yes. Absolutely. Absolutely. But where you'll get the aerobic is after 10 minutes. So if you do 2,500 steps and you do it in 5 minutes, apparently you get the best result after the 10th minute. So I would say don't be so concerned about the steps until the end of the day. Get your 11 minutes where you are puffy, you are able to talk, but you're a little breathless. If you really want to, count your pulse and get it up to, I don't know, I think it's 100 that's considered aerobic.
Good. Thanks for that helpful advice. A question for Dr. Clee now. The question's about the genetics of obesity and the obesity genes, and how did these things get created in the first place? So if someone doesn't really have a family history and their parents become obese for whatever reason, does that then mean they've got the obesity gene and it's going to get passed down and their children then are destined to become obese?
So, as I mentioned, genetics of obesity is really complicated. And what we're understanding is that the environment is playing a big role. You can think of the environment as sort of unmasking what might be genetic susceptibilities. So when you're young, you have a young family, maybe you are out exercising more, and as you get older, you're maybe less active, having a poorer diet. That could then change in environment and lifestyle, might unmask pre-hidden genetics risk factors. So, in some senses, getting obese later might mean that you're passing on genetic susceptibility, or it could just be purely environmental. What we do know is because it's due to so many factors that each have a little bit of an effect. I mean, it's not like the or those obese mice where you're passing on a gene for obesity. For most of us, because it's so many, you really can't think of it as, oh, are you passing on a risk to your kids? If you're overweight, especially when you've been younger, that's more genetically driven, so you're more likely to be passing on a higher risk, but there's no set, and you can very easily have examples where someone in a family is overweight, but their sibling is not, and that's just some lifestyle, but also the luck of which genes they happen to inherit. So did that sort of get at whoever's question, hopefully?
Good. Thank you very much.
Okay.
A question directed at Dr. Johnson, and about those interesting mice where he did the takeaway study and reduced their insulin levels. So you'll recall he showed that he reduced the insulin levels, put them on this high-fat diet, and they were protected remarkably from getting obese. And the question is, in that situation, so those mice with lower insulin put on the high-fat diet that are protected, did they get insulin resistant? Like we've been told that a high-fat diet will promote insulin resistant. What about these mice that have the low insulin?
Yeah, so that's a great question. Actually, in that study under those controlled conditions, we didn't see any sort of striking insulin resistance. So that doesn't mean there wasn't maybe a small amount at certain times in the mouse's life, but it didn't appear at that time to be a real major factor in that. So there are certainly interplays between the amount of insulin produced and the body's ability to understand it. And right now we're actually doing quite a few studies to try to tease apart that relationship. And that work's ongoing. It's actually a really interesting question.
Yeah, and so related to that, aside from the animal models, let's now talk about the human situation. So how often are we measuring insulin levels in humans and measuring or trying to determine whether they have insulin resistance? So, we're often talking about measuring glucose. We talked about hemoglobin A1C. What about measuring insulin resistance to try to get a handle on, is it the good, the bad, or?
I mean, I can start just from research, but then you can go to the clinical side of things. So, there are major studies in human populations that measure fasting insulin levels. Now, you can punch that into a formula called the HOMA formula, and you can get an index of insulin resistance from that. But in fact, those formulas, they have these assumptions that are based on correlations, and so it's difficult to know without doing a direct test of insulin resistance, although there are some labs that can do these tests. In these large population studies, what they're really measuring is elevated insulin and assuming that it's because of insulin resistance. So I think it depends which studies you're talking about. There are some studies where you can measure insulin resistance directly, but in the really big population studies, they're assuming insulin resistance from a fasting insulin level. So in that regard, in the study side of things, I think people have to now be a little bit more careful in the assumptions and do more detailed studies. But I'll pass it along to find out what they do with real non-clinical studies. So
Tom, the question is, how often in your patients are you having an insulin measurement?
I never measure insulin levels. That's not because I don't want to know what the insulin level is. It's just that the answer won't change my treatment. So why would I make it more complicated? Life's already complicated. We want to keep it simple, don't we? So the moment I think measuring it is going to change the way I manage my patients, I'll be measuring it.
Good. Thanks very much.
I'll just say that's partially because we still don't know what I showed what I think is maybe an interesting mouse study, but we still have to do the same types of studies in humans. I'm not confident that I can give him the information that would change his clinical practice. What we need to do is more research to understand the links of these pathways in human populations. And once I'm confident that we could give the clinicians advice that would change their clinical practice, then you can expect them to measure it. So I think that's a fair answer.
Jim, what is it about those skinny mice with not much insulin? Why do they not gain weight? What's happening in those mice?
You want me to talk about the browning of the white fat. So I went over it a little bit quickly. Now these are questions from the panel to the panel. So
who's interested in brown fat? That's what bears hibernate with.
Yeah, so most fat, the majority of the fat we think of as fat, its job is to store energy. But there's a special type of fat that's packed with these energy powerhouses, these mitochondria, so much so that it actually looks brown. And it used to be thought that only bears and other hibernating creatures had it, but now we know that human beings have small amount but a potent amount of brown fat. and it wastes energy. It turns energy into heat, literally burning calories away. So one of the observations in our study is that the white fat, which its job is to store energy, store as much as it possibly can, it got the tiniest bit brown. And you could imagine over a long period of time that tipping that balance between storing energy and burning just a little bit would be in favor of getting rid of some of these calories. So I think what Dr. Elliott's trying to get at is we need to do more research to understand if we could get, if we could modulate the browning of fat or the amount of brown fat in humans through interventions such as diet that might affect insulin levels. Because if you could do that, if you could make concrete recommendations, and maybe a certain person's genetics would be susceptible to this, but in other persons it might not work. So again, that's the personalized medicine. So if you would be a candidate to have your white fat turn brown and melt, literally, now I'm going to sound like Dr. Oz, but it would, would literally burn off energy. So the answer is that right now it's very fanciful, but theoretically these things are theoretically possible.
So that brings another question related to the kind of timing of these things. So we're talking about personalized medicine, personalized genetics to treat obesity. This question was specifically about obesity. How long do you think some of this research is going to take to move forward into personalized therapies for obesity?
So I think there's some things that could be done very quickly. So there's sort of the tip of the spear. There are things that we have a pretty good handle on that, like the biomarker questions. We know, for example, there are some drugs that work and some that don't. And I don't think it would take that long if we were able to get a handle on people's genetics. It used to cost, so to sequence the first human genome took hundreds of millions of dollars and about a decade. Now you can sequence a human genome in a lab for $1,000 in a day. So, if I had a decent-sized grant, I could sequence all your genomes and find out how you responded to the different drugs and the different diets. And this is, in fact, what we're proposing to do. So open your checkbooks, but and send us some genome. But anyway, the point is some of those things are actually not so crazy. The information, the actionable things, the drugs are there. Some of the other things, such as turning white fat brown and other things, and developing new pills that could do some of these things. Of course, these are quite far off, and I don't like to make timing things. I just can tell you, it's not coming tomorrow. I don't know if you want to add. Anyone else?
Maybe Dr. Clee to add to that, but just before she does, note Wednesday, February 4th in your calendar if you're interested in this topic, because the LSI is sponsoring another workshop, the title of which is Preventative Health Molecular U, and it's going to talk about some of these issues of collecting your DNA and sequencing it and using that potentially to give you some more information about yourself and the sorts of medicines you may or may not respond well to. So that, I think, will be a very provocative talk Wednesday, February 4th. Dr. Clee, what's your forecast of when we're going to have some
the magic pill for obesity?
Based on genetics.
Unfortunately, obesity, because it's so complex, we're really lagging behind. For diabetes, we'll be there much sooner because even though it's still complicated, it's far less complicated. So there are things already we know, things in the works, like trying to understand which drugs will work better for which people. For obesity, the unfortunate answer is we just don't know enough yet. So we don't have understanding of what genes are involved and how they work to be able to start making those predictions. Now, hopefully, over the next few years, some of that may start to come to light. The human genetics is getting there, but all of this research takes time, and like those human genetic studies I was talking about that involved over 300,000 people, you can imagine they're very expensive, and so the research is progressing as quickly as it can, but for obesity, I don't think in the immediate future, like Dr. Johnson said, it's hard to predict and really put a timeline on things, but that's still a ways down the road.
Let's make one small comment. The research is progressing as fast as it can with the people and the resources we have. But events like this, where hopefully the people and the public go away thinking about research, we're in a time now where governments are starting to think, well, do we think research is valuable or not? The economic case is there to do this research. But, all across the world, one of the first areas of budgets that get cut is the research side of budget. So as voters and as the public, I think it's important to understand the research is going as fast as it can with the resources we have. But all across Canada and all across the world, lots of scientific labs are being shut down or having their funding reduced, so that it's only going as fast as the public wants it to go. So I'd encourage you to think about these things as well. So we're trying best we can, but it's tough times as well.
So that's a good segue into a related question. You kind of talked about research around the world, and this specific question said, okay, we talked about some of the research we're doing at the Life Sciences Institute. Is there similar research being done at other institutes around the world, Europe, for example? And what about pharmaceutical companies? Are they involved in this? What's happening outside of the University of British Columbia?
I'll start this one off because I'm actually on a sabbatical right now, as was mentioned. So I'm in Oxford for the year. And some of the things that we do really well here, they don't do over there. But what they do really well is actually these human studies. So they are really pioneers in what's called the genome-wide association studies, which is where you make these huge lists of these hundreds and thousands of genes that affect different traits like obesity and height. And I have it on good word that a series of major papers are going to come out soon on the genes that affect fat distribution and how obesity shows up in different parts of the body. So some of these, there are big breakthroughs happening all over the world. It's a worldwide thing. I don't know if you want to add to that.
Pharmaceutical companies get a bad rap, to my mind. The pursuit of profit by the shareholders is actually driven by excellence in drugs, and the expensive drugs we have to pay for have allowed science to progress rapidly. I did it out in Burnaby on Enterprise Avenue. Is a huge research institute owned by Amgen. Amgen is a company that makes medication for arthritis. Actually, it's made from mice. They get antibodies from mice, and they target the problem that causes arthritis, or they target certain cancer problems. They've got a new medication for cholesterol. Right here in Burnaby, it's a very impressive laboratory. So there are lots of applications, and there is a price, and we pay it at the pharmacy, but the benefits we're reaping are extraordinary. Extraordinary.
Good. Thanks very much. I just want to move on with one last question.
Do you want to comment on your collaborations with the companies? No?
Yes, we do. Come on.
Let me ask a question to follow up with Tom's, and I promised people would get out by 8. So you talked about antibodies being raised in mice, and we heard today some research from Dr. Clee with mice and Dr. Johnson with mice. And the question is, are there alternatives to using mice in medical research, such as human cells? So I think it's a good way to finish our discussion. Why do we use animal models for research, and are there alternatives? Are we using those as well?
So I'll talk a little bit about obesity first. And, of course, you can imagine that, yes, we can study obesity in people. But like I was saying, because your body weight is so related to how much exercise you've done over your life, your diet, your emotional state, your stress levels, all these different things that we've heard about, that makes it really, really difficult. And because of all those different things that can sort of as a side effect influence body weight. When you try and look at any one thing, whether it's a diet or anything else, you've got all these other things that are tweaking body weight. And so, like Jim was mentioning, to do the studies that we really need to do to look at the effects of diets require just enormous numbers of people and cost astronomical amounts to be able to do. So what we really feel for the obesity, because it's so complicated, if we can use mice or any other model system where we can control a lot of those things, keep them all constant, and really hone in on one thing at a time, and then take that and know what to measure and what to look at in those studies can be far more effective later. But obesity is complicated. It's not just one tissue or something you can look at in a cell. It's how your liver's working, how well your digestive system's working, how well your pancreas is producing insulin and other tissues that produce different hormones. Your brain actually controls a lot of your food intake and your energy expenditure responses. And so the only way to really be able to link those all together and understand how they all work together is in some sort of a living, breathing animal that has all of those tissues and organs.
So, Jim, I'll give you the last word, but maybe include in your answer, how are these animals treated? Is there any regulation? Is there any oversight? Please comment on that.
That's a great question, and I think a lot of us think about this a lot. I got into biology because I was interested in animals and humans, of course, but how they work. And I think a lot of the people in the field start off with that sort of wonder. One of the key things I mentioned in my first few slides is the ability to do these powerful loss-of-function experiments. And I can tell you that it's not possible. Forget about convenient and costly. It's just not possible for me to take one of you and delete some of your genes. Nor would you probably want me to try that. So there are things that, there's really no other way around trying to do it. And we think about these things all the time. The other thing, as Dr. Kiefer is alluding to, is the mice we use are, they live very, very pampered lives. So the facility is fairly incredible. I sometimes wish I had the life they have. They eat ad lib. They have vets watching over them at all times. We go through incredible amounts of regulations, certifications to be able to just have a mouse, not to do anything to it, to have one. I can tell you there are a lot of different model systems that people use. People use mice, fruit flies, worms, fish. The fish, for example, Dr. Kiefer has zebrafish, which are this really cool genetic model in his lab. And I can tell you they're better treated than most goldfish in kids' goldfish tanks, which, as we know, it's never one goldfish that lives the life of the kid. It's 12, and they get replaced, and they go down the toilet. So anyway, I'll just a little bit of a joke there, but, we are committed to doing this research because we think we're going to answer fundamental questions that are going to help human beings. It's human beings that are paying for the research. They go out and raise money, kids who have type 1 diabetes, people who have type 2 diabetes, people who've lost limbs, who've gone blind. And, of course, you have to do some of these studies, and we think a lot about them. But at the end of the day, we're really committed to trying to cure these diseases.
Good. Thanks very much, Jim. So I'd like all of you to join me in thanking the speakers for a very excellent series of talks.